Again, the only reason I have this image on here, this image doesn't show up in your book, is to give you the idea that this photorespiration is dependent on the mitochondria in the plant. So you have some similar players in here. You have your rubisco. Remember, the rubisco is what's going to be responsible for carbon fixation. But in um, photorespiration, we don't have carbon dioxide because the stromata are closed. This is because we don't want to lose water. So the thing about rubisco and its ability to use oxygen, they think this is a revolution or an evolutionary relic, because rubisco has always been able to use oxygen, but it was never a problem when there was very little oxygen. When it was first evolved, it evolved in a climate where there was an abundance of CO2. So the fact that it has an affinity for oxygen wasn't an issue at that time, but in this scenario, it becomes an issue because suddenly there's a lack of CO2. And that's one theory. Another theory is that it evolved to limit the damage of light products or light reactions in the absence of the Calvin cycle. But at any rate, with the C3, C4, and the CAM plants, each of them have adaptations. The C3, the least adapted. And by the way, that C3, that 3 comes from how many carbons go into the original Calvin cycle. There are 3 carbons go into the C3 cycle, 4 into the C4 plants, and then the CAM plants are so named, and we'll find out why that's named that way. But what happens here is you actually have kind of a byproduct that goes through a peroxisome and then into the mitochondria. And many problems with this is because it drains much of the fixed carbons. It also uses ATP. C4 plants adapt and minimize, and the C4 is because of the fixed carbon product. They minimize this by incorporating CO2 into a 4-carbon compound in the mesophyll cells. We're seeing this here. If you look at the far right, the top of the far right, that's the mesophyll cell. You have a enzyme called the PEP carboxylase, and what that can then do is it turns CO2 into this 4-carbon product, and that then can go into the Calvin cycle. It's a temporary product. It's malate. That 4-carbon can go then into another portion of the plant called the bundle sheath cell and be split apart. You have pyruvate, which is a 3-carbon, and carbon dioxide, which then can go into the Calvin cycle, produce sugar, which we're familiar with. Go into the Calvin cycle just like we think of it before. And this particular arrangement actually separates the two between 2 different parts of the cell of the photosynthetic area of the plant. You're seeing the image over on the left where you have your mesophyll cells and then your bundle sheath. The mesophyll cells are on the outer portion. You have your vein in the middle, the vascular tissue. The mesophyll cells, the outer portion, the bundle sheath on the inner portion. So 2 different areas of the C4 plants. And here's a little bit of a compare and contrast between C4 and C3. C4, the organization between the mesophyll and the bundle sheath cells, a little bit more organized. You have the same thing happening you have the same thing happening in the C3, not quite as organized, but you still have the capacity for this particular arrangement to take place in C3, just not quite as adapted, quite as much adaptation. Just the most important thing with regard to this is you don't have to memorize all these chemicals or even be overly familiar with all the chemicals, but that the 4-carbon compounds are exported into the bundle sheaths, where then they can then be released as CO2 that can go into the Calvin cycle. And then this plant can doesn't have the buildup of or the reduction with regard to CO2, reduction of carbon dioxide. Some plants, like succulents, and if any of you have ever grown succulents, they don't need very much water at all. They use a particular compound called crassulacean acid, which we very kindly get to abbreviate it as CAM. That's why we call them CAM plants or CAM plants. To fix carbon. They actually even operate slightly different. They open their stomata at night where there is less chance of losing water, and incorporating the carbon dioxide that they open those stomata for into organic acids, and that then they close during the day. And then that stored form of CO2 is released from the organic acids and used in the Calvin cycle. And a type of CAM plant is pineapple or succulents. I'm sure you all know what a succulent is. A type of C4 is sugar cane. And a couple of the things that they're doing is they're trying to adapt C3 plants into more of this C4 arrangement to make them more water sparing. And then they're also looking to see how, if the climate changes, how that will affect the ability for each of these types of plants to adapt. A couple of other important things for you to have in your head. You have separation of steps in the C4 between the mesophilic cell and the bundle sheath. You have a time separation in the CAM plants between night and day. So those become things to maybe have in your head when it comes to test time. Then the last thing we need to talk about is the whole process of this whole process, and why is it important. So the important reasons why we're learning about photosynthesis and why it's important to study. So the importance of it is the chloroplasts have the ability to capture energy from sunlight and store it as chemical energy in organic compounds like starch granules. You see them here. Sugar is made in the chloroplast, supplies chemical energy and carbon skeletons to synthesize organic molecules of the cell. So that sugar that's made by the plant then can become more of us or any other heterotroph. The plant can store that sugar as starch in structures such as the roots, tubers, seeds, and fruits. And in addition, photosynthesis is very, very important with regard to putting oxygen back into the atmosphere. So in addition to food, photosynthesis is a very important process because it creates oxygen for us to breathe. It's the other side of the coin. We create plenty of carbon dioxide for plants to use. Plants create oxygen for us to use, as well as this stored form of organic molecules. Any way you look at it, you can call it sugar, you can call it starch, but it's a stored form of organic molecules that even if you're not a plant eater, you eat something that eats plants. So somewhere along the line, you may be saying, I don't like plants, I don't like vegetables, but you're eating something that does like plants or vegetables. So that's why we study photosynthesis, and we'll find out how we study the next topics that we go through. See you soon. I forgot to add this piece to it. I forgot the last one, and here you have what you need to be able to more or less talk about. Hopefully I didn't mess this whole recording up. But describe the structure of the chloroplast. I think you can probably do that. The relationship between action spectrum and absorption spectrum. Trace the movement of linear electron flow as well as cyclical electron flow. And then describe the similarities and difference between oxidative phosphorylation, that's what happens in the mitochondria, and photophosphorylation in the chloroplasts. The role of ATP and NADPH in the Calvin cycle, and major consequences of photorespiration for the plant, and two important photosynthetic adaptations that minimize photorespiration. And now we are finished, and we will start with the next chapter.